Hi, it's Mr. Andersen, and in this podcast, I'm going to go over non-Mendelian genetics, or sometimes refer to this as everything that Mendel didn't know. And so I'm going to kind of build on the work of Mendel and then eventually get into the work of somebody named Thomas Hunt Morgan. And so the first few things are going to be just kind of terms that you should understand. First one's going to be incomplete dominance. Incomplete dominance sounds a lot like blended inheritance, but basically what happens is, let's say this one's big W, so this is white snapdragons. This one's red. If you cross the two, so our hybrid, when we get one gene from each, is going to be pink. And so red plus white equals pink. Now, if we were to really go back to blended inheritance, you would cross pink and you'd keep getting pink. But on incomplete dominance, if we were to cross pink with pink, and we do the Punnett square for that, like that, what we're going to get is a red snapdragon. We're going to get a pink snapdragon, and a white snapdragon. And so we can get that white and that red back. And so incomplete dominance is when if you get one gene from each of these two, then the combination is going to be somewhere in between the two. Now, something similar to that is called codominance. In this one, if you've got heterozygous for red and white, you're going to have splotches of white and splotches of red, like in this rhododendron or in this roan cow. If we have a red and a white cow, then the offspring is going to be red and white. But it's not like pink. That would be cool to have a pink cow. But it's going to be hairs that are red and hairs that are white. And even in these red areas, you pull one hair out, it's going to be red. One hair out, it's going to be white. And so in codominance, we're basically going to have splotches of the two different colors. But the genetics are exactly the same as incomplete dominance. Another term is called epistasis. And so if we were to look at this alligator, what color is its skin or what color is this guy's hair? Well, we don't know because there's a number of genes that probably determine the color, but there's going to be a separate gene that determines whether that actual color is added to the hair or not. And so if there's a problem in that gene, then it's going to result in albinism. And so basically epistasis, what you should remember, is that there are certain genes that can affect a number of other genes, almost like an on-off switch. Without that on switch, the hair doesn't get added, and so you have an albino. Sometimes it's hard to do genetics problems dealing with epistasis. Sometimes we'll have multiple alleles for a trait, and so an example of that is going to be blood type. So you could have the A protein, you could have the B protein, you could have neither protein, so we get the 4 different blood groups. I'll make a separate video because this is sometimes confusing and explain those, but know that we've got, instead of just 2 choices, we've got, in this case, 3. Next one, as we get into Mendelian genetics, we start to realize as scientists were doing work is that some of the things that Mendel had discovered didn't seem to be holding true. It was almost as if he lied to us. And so we call these non-Mendelian genetics. Whoa, that's scary. So the guy I want you to remember here is Thomas Hunt Morgan, and he worked with fruit flies, and he figured out some pretty amazing things. And this was, we kind of had discovered the work of Mendel, but also they had discovered these things called chromosomes, and we were starting to kind of work out how that comes together. And it was convenient because each of the traits that Mendel had studied, so flower position, color, pod shape, all of them were found on separate chromosomes. And so he never had a problem with what we call linkage between genes. And probably if he ever did do traits where the 2 traits were on the same chromosome, he probably didn't include that data because he got weird numbers that just didn't fit. We know that some genes flow together, like freckles and red hair, and that's because those 2 genes are going to be found on the same chromosome. This is the experiment that showed Thomas Hunt Morgan how this works. And it's kind of stick with me. It's a little bit confusing, but I think you can do it. And so basically the way we name our genes in fruit flies is going to be a little different. We basically find the mutation. In this case, you can see that this Drosophila is black, so it's got this BL mutation. And then it's got these weird vestigial wings, and so we call that the VG mutation. The normal run-of-the-mill fruit fly is going to be, we just put a plus next to it. And so this would be the wild type, we call that a fruit fly. And so if I were to really write this out using terms like you're maybe more familiar with, a better way to write it might be like this, V little b, and then we cross that with little v, little b, little b. But get used to this because it shouldn't be that confusing. And so basically what he did in this cross is he took one fruit fly that was, let me go back to that. One fruit fly that was heterozygous for the 2 traits and one with homozygous recessive. And so if you do the Punnett square, this one is going to give a VG, vestigial gene, and it's also going to give a black mutation. And so really this one can only give one type of gamete. But the other one over here, so if we look at the other parent, so this mom right here, it could give VG plus, so it could give this VG plus, and it could give BL. But it could also give VG plus, BL plus. It could also give a VG BL plus, or it could give a VG BL. So it can give those 4 different combinations. And so this one right here is going to make one that doesn't look like the parent, but this one would look like the parent, and this one would look like the parent as well. And so basically, according to Mendel, you would get 2 of them that look just like the parent, and then 2 of them that are going to be combinations. Like this one right here is black, but it's got good wings. And this one's not going to be black. It's wild type, but it's got these vestigial wings. And so we call these things recombinants, or a recombination of the 2 parents' phenotypes. And so Thomas Hunt Morgan's crossing these fruit flies, and Mendel says that he should get 50% parental. You can see it, 50% parental, these 2, and then 50% recombinants. And so he knew his genetics, and he knew he should get 50% of each, but he didn't. Time after time after time, he just kept getting 17% of the recombinants over and over and over again. And so he couldn't figure that out. Why is it 83% of these and then 17% of these over and over and over again? They were starting to think maybe they discovered some new number like pi 17 and its significance. But sometimes in other crosses it'd be like 9%, some it'd be like 21%. And so basically one of his students, Thomas Hunt Morgan's students, Alfred Sturtevant, went home one night, blew off his homework, and kind of figured this out. And what he figured out, let me clear this out, is that there are going to be chromosomes. So the chromosomes look like this. And some genes are going to be found on the same chromosome. So we might have the BL mutation right here, and we might have the VG mutation right here. And since they're on the same chromosome, they're going to follow each other through generation after generation after generation. And since they're tied together, we're going to have more of the parental types than we are of the others. And you might think, well, how do we even get recombinants if they're tied together? Remember, during meiosis, we have crossing over where this will actually split. And so there'll be crossing over between chromosomes. And so the cool thing is if those 2 genes are really close together on the chromosome, it's rare that they're going to be split in half during crossing over. And so 17% told him that they were, if they're on different chromosomes, we would get 50%, but anything less than 50% is going to tell us how close they are. And so let's say the frequency of recombination is 3%. That means that those genes are going to be, there might be another gene right next to this one. And since they're so close on here, the odds of them being split in half is going to be really, really rare. And so what he discovered is this idea of gene linkage, that there are some genes that are found on the same chromosome, and they travel together. Another thing that we're starting to discover is that most traits are caused by multiple genes. The standard that biology teachers will say is that you've got a hitchhiker's thumb, and it either is a hitchhiker's thumb or a straight thumb. But it's probably not that simple, because when I talk to kids in class, you'll find that some are really straight and some are in between. And that's because most traits, almost all traits, are caused by multiple genes interacting. And so if you flip 2 die, what are the odds that it's going to come up snake eyes or double ones? Well, there's only one way to get that, a 1 here and a 1 here. And so the odds are 1 in 36. But there are going to be some numbers that come up all the time. So if we look at that, 7 is going to come up all the time, almost 1/6 of the time. And so if you ever see a bell-shaped curve in phenotypes, physically what they look like, that means that there are multiple genes taking place. In this case, it would only be 2 genes, and we can get this nice bell-shaped curve. So let's go to something that we're all interested in. That's going to be how tall you're going to be. Basically, if we go to the simplest level, height genes, let's just make it simple and say that there are 6 different heights that you can have, and that's contributed by different genes that you get. And let's say we've got Jimmy here and Susie, and Jimmy and Susie meet each other, but then they get back together later in life and they decide to have a family and they decide to have a bunch of kids. Let's say they're of average height. We'll call them height 3. And that means that they've got 3 different genes, but they're heterozygous for each of those. So they've got a tall gene and a short gene, so they're heterozygous for each of those. And so since they have 3 tall genes, we'll say that they're going to be of average height. But now let's work out the Punnett square with just their genetics to figure out how tall their kids might be. In other words, Jimmy might give a big A, a big B, and a big C, and she might give a big A, big B, big C, and so they'd have a sixth child, so going to be a giant kid. Or they might give all their short alleles, and they're going to have a really short kid. And so if we start punching out the numbers, what we find is that the odds of them having a really small kid, we'll call them height 0, is going to be a 1 in, this would be 64 chance. But as we work our way up to 2s and 3s and 4s and 5s and 6s, what we'll get is that characteristic bell-shaped curve. And so it's going to be a bell-shaped curve like this. Most of their kids are going to be of average height because they're of average height. Doesn't mean they can't have a really tall kid or a really short kid. It just means that they're probably going to have kids that are in the middle. Now, how many genes actually affect height in humans? Dozens and dozens of genes. And so you can imagine that we're going to get a huge bell-shaped curve. And that's why we have a bell-shaped curve of life. Everything from the length of our thumb to the length of our foot to our intelligence, all these things are caused by multiple genes. And so it's not as simple as a simple Punnett square. And then the final thing that Thomas Hunt Morgan added to the work of Mendel was the idea of sex linkage. And so he was doing crosses where he would cross flies that are going to have this white eyes with those that are the wild type. And he'd find that in the F1 generation, they're all going to be the same. But when the F2 generation, only the males would have the trait. And so what he discovered was sex linkage, that there are some traits that are genes that are actually found on the sex chromosome. In humans, we see this in color blindness. So if you look here, there should be, and the color's always hard to hit, but there should be 3 numbers that you can see in each of these. And if you can't see any of these, you might be colorblind. 37, 49, 56. And I'm colorblind, so I struggle with all of those. So how does that work? Well, basically, we have some traits that are carried on the X chromosome. And so let me show you how this works out in my family. Basically, this would be my mom. She is XX, and she's a girl, but she has a mutation on one of her X chromosomes, or a faulty gene. And so if I were to punch the Punnett square, my dad is not colorblind. So if I do this, this is going to be XCX. This is going to be XCY. This is going to be XX, and then this would be XY. And so you should be able to understand what each of these are. This would be a normal female. This would be a normal male. This right here is going to be a colorblind male. So we could say this is me. So I got one X chromosome that's got a mutation. My dad gave me the Y chromosome, and so I'm colorblind. If we look at this one's going to be a carrier of colorblindness. And so this could be my sister, because my sister's not colorblind, but that's because she has one good X chromosome. How do I know that she's a carrier? Well, I have 2 nephews that are colorblind, and so she passed this faulty mutation onto them. Their dad, who's not colorblind, gave them the Y, and so then they ended up being colorblind. And so it's a bummer to be a boy sometimes, because we only have one gene on our X chromosome, and so if it's faulty, then we're going to become colorblind. Now, in human population, 1 out of every 10 genes that cause this color deficiency are going to be faulty. And so boys, since we only have one X chromosome in our population, 1 out of every 10 males are going to be colorblind. So you may be able to figure out in your head, how often is that going to happen in girls? Well, since they have 2 X chromosomes, it's going to be a 1 in 10 probability on the first X, and it's going to be a 1 in 10 probability on the second X. And so it's going to be a 1 in 100 probability that girls are going to be colorblind. And it's rare that I find girls in my class who are actually colorblind. And that's because they have, the extra X chromosome, that backup copy, which we do as well on all the other chromosomes inside our body, since we're diploid, except the sex chromosome. Another trait that is common here would be hemophilia. Hemophilia is one that's carried on the X chromosome, more common in boys than girls. But I love evolution. Whenever I see a number like this, 1 in 10, that seems like a high probability. Perhaps there's a evolutionary advantage to being colorblind. And I've read that if you're colorblind, when you're hunting or when you're in the military, you can actually pick up camouflage better than you can if you have regular vision. And so maybe there was an evolutionary advantage at some point for people who were hunting, especially hunting in and around night, that they could maybe see better and were more successful. And that's why I love biology. You'll never know all the answers, but you sure know a lot. And so that's non-Mendelian genetics, and I hope that's helpful.